Hello, everyone. I'm Yuxiao Yang, and I'm an assistant professor in the Department of Electrical and Computer Engineering, as well as the Disability Aging and Technology cluster. So first of all, I would like to thank the CECS Seminar Committee for providing me this opportunity to talk about my research. And today, I will talk about next generation brain-machine interfaces, or BMIs, for control of brain states. So a bit about myself. So I joined UCF this fall, and I obtained my Ph.D. in electrical engineering from the University of Southern California in December last year. So my research focuses on developing closed-loop BMIs for neuron coding control and spans systems and control theory, machine learning, and neuron science. So my teaching primarily focuses on systems and control theory. And today, I'm going to talk about my research during the past few years, mainly done during my PhD, and parts of my research have been published in Nature Biotechnology and Nature Biomedical Engineering, and has been selected as the cover article. So let's start from a big picture of my research. So a brain-machine interface basically creates a direct control pathway between the brain and an external device. So in the past decades, there has been much progress in developing BMIs that use the brain to control machines. So in this type of BMI, typically, we first collect electrical signals from the brain and then interpret the subject's movement intention from the brain signals and use it to control an external device, such as a robotic arm, to execute the movement. So in this BMI, the brain is usually modeled as the controller and the external device is modeled as the plant to be controlled. Such BMIs hold great promise for treating paralysis, for example. However, in many other medical applications, such as treatment of depression, we need to control the brain state itself, such as mood, using external stimuli, such as electrical stimulation. So this calls for a new type of BMI that actually uses the machine to control the brain state itself. In this BMI, the aim is to control the external stimuli to regulate the brain states to be at a healthy level. The key is that the delivery of the external stimuli is guided in real time by brain signals, thus forming a closed-loop system. Now, the brain is the plant to be controlled, and the goal here is to design the controller. So this new type of BMI has applications in treating, for example, neurological disorders such as Parkinson's disease, neuropsychiatric disorders such as depression and PTSD. It can also be applied to modulate unconsciousness states under anesthesia, and it can also promote scientific discovery. So in the next few slides, I will talk about how I design and validate the key elements in this new type of brain-machine interface using tools from systems and control theory, machine learning, and neuron science. So to control the brain state, we first need to decode or estimate the brain state from the brain signals. So in this slide, I will introduce how we decode an important brain state, that is mood, from brain signals in human subjects. Mood decoding has not been achieved before due to two main challenges. First, the mood encoding network is distributed across multiple brain regions, and the mood encoding network is not well understood. So this requires recordings from multiple brain regions and will lead to high-dimensional neuron features. Second, mood is hard to assess, and this leads to sparse mood measurements. So therefore, on one hand, we have high-dimensional features. On the other hand, we only have very sparse training data for machine learning. And this creates a challenging problem. So to solve the challenges, so in collaboration with UCSF, so we recorded multi-site intracranial electrocardiogram signals from 7 epilepsy patients continuously for days. And at the same time, we used a self-report questionnaire termed immediate mood scaler to measure their mood a few times a day. And then we use dimensionality reduction and machine learning to train the mood encoding model to relate the brain signals to the IMS. Once we have this mood encoding model, we use the trained model to predict the mood from the brain signals in the test set. So we successfully decoded mood from each subject in cross-validation. So here I'm showing the true mood measurements, which are the questionnaires we obtained from subject in the x-axis, And the y-axis, I'm showing the predicted mood by only looking at the brain signals. So the cross-validated prediction, they fell along the 45-degree line, showing good decoding performance. And this good performance is achieved in each and every one of our subjects. So to summarize, we successfully decoded mood from brain signals, and this result can guide clinical diagnostic and closed-loop treatment of various neuropsychiatric disorders such as depression, anxiety, and PTSD. And this work is also selected as the cover article in Nature Biotechnology in 2018. However, even if we can perfectly decode mood, in order to control it, we still need to know how electrical stimulation affects the brain signals. Predicting the brain's response to electrical stimulation is challenging because the brain network organization is not well understood, posing a black box modeling problem. Also, we usually have high-dimensional brain signals, but only limited data for training, again making the effective learning of the model difficult. To solve these challenges, we developed a data-driven method to model the effect of stimulation on brain signals. Basically, we applied a specific stimulation waveform to the brain, and at the same time, we recorded the brain signals and then trained the model to relate the stimulation to the brain signals. In particular, we developed a binary noise modulated waveform that is both clinically safe and theoretically optimal in eliciting the informative brain response for machine learning. So in collaboration with NYU, we validated the modeling framework using intracortical stimulation, electrical stimulation, and intracortical local field potential recordings in monkeys. So we find that our model successfully predicted the brain response to electrical stimulation. So here in test sets, I'm showing the ground truth brain signals in gray here, and I'm showing the model predicted response in red. So we see that our model achieved good prediction of the response in various brain regions, as I indicated by these dots in these two monkeys. So to summarize, we predicted the time evolution of the brain response to electrical stimulation. The results give insights into the dynamics of the brain response to stimulation and constitute another key element in enabling closed-loop BMIs for treatment of neuropsychiatric disorders. And this new work has just been accepted by Nature Biomedical Engineering. So now we can both decode mood and also model the stimulation effects. So the next step is to combine these two elements and then build a closed-loop controller that can take the estimated mood as feedback and then optimally adjust the amount of stimulation I deliver to the brain so that I can regulate the mood state, for example, for the brain. And this is future work, and I will touch on it more later. So the third topic is about developing a closed-loop BMI for automatic anesthetic delivery. So the motivation here is that manual anesthesia, meaning maintaining anesthesia by an anesthesiologist in the ICU or the operation room is usually imprecise and costly. So automatic anesthesia can thus improve clinical efficacy and efficiency. So therefore, we developed an adaptive closed-loop BMI to control a particular type of anesthesia or anesthetic brain state that is termed medically induced coma. And medically induced coma is a very deep anesthetic brain state. So in collaboration with MGH, we tested the adaptive closed BMI in rats. So we show that we achieved tight control of the depth of anesthesia. So here I'm showing the result from a real-time experiment in one rat. So the first panel shows the brain signals, which were used as feedback to adjust the anesthetic infusion rate. The second panel shows the controlled levels in black, the controlled level of the depth of anesthesia, whereas the target level is shown in green. The third panel shows the controlled anesthetic infusion rate. So we see that the adaptive closed-loop BMI maintained the depth of anesthesia precisely at its target levels and achieved prompt transitions without over or undershoot. So to summarize, our results have implications for automatic anesthesia care in the intensive care units and can give insights into the pharmacokinetics and pharmacodynamics of anesthetic drugs. So next, I will briefly talk about my plans. So for the near-term plan, I will focus on algorithm development for nonlinear closed-loop BMI systems and also preliminary data collection using high-density EEG system. I plan to collaborate with the ECE, MAE, and also the biomedical sciences at UCF. I have already started a project with Dr. Amen Bihao and Dr. Chu Shifu to build a real-time BMI system that integrates EEG and robotics. I also aim to seek for other collaborations on building BMIs for decoding and understanding various cognitive functions. So in terms of teaching, I've already designed and submitted a new graduate course on probabilistic methods for neuron coding and control. For the next 5 to 10 years, I would like to build non-invasive BMIs for treatment of neurological and neuropsychiatric disorders. So the closed BMI will integrate non-invasive EEG recordings and also non-invasive transcranial magnetic stimulation. So non-invasive BMIs do not require brain surgeries to put in electrodes inside the human brain, so it can benefit a wider patient population. So I will develop new closed-loop control algorithms that are suitable for non-invasive BMI systems. I will conduct human experiments both at UCF and also in collaboration with local hospitals. I also want to move toward personalized BMIs for anesthesia care. So I plan to collaborate with the biomedical sciences to conduct rat experiments and collaborate with local hospitals to conduct human experiments. And of course, I will also aim for tech translation for the clinical use of the above two systems that I have envisioned. And finally, I would like to thank my PhD advisor, Professor Mariam Shonichi, my collaborators, the funding resources, and thank you, and I'm happy to take any questions.